And hi, everybody. Welcome to today's science seminar. I am Jess Delfiaco. I'm the communications manager for the Midwest Climate Adaptation Science Center, or the Midwest CASC, as you'll hear us call it. A few housekeeping notes before we get started. We are recording this seminar. It'll be available via email and on our YouTube page shortly after today. So please keep your own video off until we get to the Q&A portion of the event, unless you are a speaker. Um, Speaking of Q&A, we are going to have a little time at the end of today's presentation for you to ask questions of our presenters. So please feel free to pop those questions into the chat throughout today's seminar, but know that you will have a chance to come off mute and ask those directly as well at the end. Who and what is the Midwest CASC? I'm guessing this is a familiar slide to many of you, but for those of you who are joining us for the first time, the Midwest Climate Adaptation Science Center is a partnership between the U.S. Geological Survey and a consortium of 9 institutions in the Midwest region, hosted by the University of Minnesota Twin Cities. You can see all of our members on that map up on the screen there. So our mission, we're part of a national network of regional CASCs, all of which work to produce science that can inform research management efforts and help fish, wildlife, water, land, and people adapt to a changing climate. We accomplish this mission in several ways, including conducting synthesis research on topics relevant to regional priorities, which are relevant at a national scale. We support a network of graduate students and postdoctoral fellows, including the 2 that you will be hearing from today. We also partner with the College of Menominee Nation to run a summer tribal undergraduate research program. And of course, through the CASC, the USGS funds many climate adaptation research projects and partnerships. So with that out of the way, I just have a couple of announcements. Today is the final opportunity to apply to participate in the Midwest CASC's 2025 mentoring program. This is a 3-month-long virtual program, which is open to all Midwest CASC affiliated graduate students and postdoctoral researchers. If you want to learn more, you can scan that QR code. I'll also pop the link in the chat to check out that short form that we will use to gauge interest. After today, we'll start making matches between grad students and postdocs and our mentors, which are adaptation practitioners from around the region. So if you are a graduate student or a postdoc, or if you are an advisor, please, just a final reminder, if you're interested in participating or you'd like to learn more, check us out at that link or reach out to me and I'm happy to chat. And then lastly, I wanted to welcome and give an opportunity to 2 of my newer colleagues to introduce themselves to folks on the call. Maybe Wes, do you want to go first?
Sure. Good morning. Thanks, Jess. Hey, everybody. I'm Wes Bickford. I'm a biologist at the USGS Great Lakes Science Center. I'm based in Ann Arbor, Michigan. And as the slide indicates, I've recently started a visiting scientist role with the Midwest CASC. So I'm really excited to get going on this project. So a little bit about me is that I've been with USGS for about 10 years. My background's in wetland ecology and in invasive species and in plant microbe interactions. And I'll be working on a project here, kind of looking back at wetland restoration efforts to learn about how well we've done setting climate-informed goals and meeting restoration objectives, kind of given climate exposure. So I'm really excited to join the group, and thanks for having me.
Thanks so much. Kevin, do you want to say hi?
Yeah, thanks, Jess. Good morning or good afternoon, everyone. As the slide says, my name is Kevin O'Donnell. I'm the new Assistant Regional Administrator for the Midwest CASC. I've been in this role for about 3 months. Prior to joining USGS, I was with the Environmental Protection Agency, EPA, in their Great Lakes National Program Office. And for 14 years or so was working on the Great Lakes Restoration Initiative in the Great Lakes, working with many federal, state, and tribal partners to protect and restore that system. So I'm very happy to join USGS and all of you in your work. And yeah, thanks so much.
All right. Thanks, Kevin and Wes. And now we are on to our main event. We have two speakers for you today. Isla Skarupa is an affiliated postdoctoral researcher with the CASC based at the University of Illinois, and Gigi Voss is a graduate student at the University of Minnesota. I'm going to hand it over to them. They can tell you much more about themselves and their research than I can do in a couple of minutes here. So Gigi, if you're ready to pull up your slides, I will pull mine down. As you said, folks, hold on to those questions or write them down in the chat, and we will get to them after the next presentation. Isla, if you want to pull your slides up, the floor is yours.
You and thanks folks for staying on after that great talk. I'll be switching gears a little bit to talk about aquatic inverts in relation to climate change. This project is fully funded by the Midwest CASC, and we started it a little under a year ago. So I'm Isla Skarupa, I'm the postdoc on the project, and the PI is Jason Breed, and we're both out of the Illinois Natural History Survey. So river ecosystems are largely determined by the natural flow regime. Flow determines in-stream habitats, which in turn determine aquatic biodiversity. And species have evolved specific life history strategies in direct response to these natural flow regimes. So unfortunately for projections in the Midwest, there will be an increase in the frequency of wet to dry and dry to wet transitions. And here's just 3 different scenarios of low, medium, and high change. And this will have direct impacts to the natural flow regime and aquatic biota. And it's also shown that by 2100, the Great Lakes region average air temperatures are projected to increase by 3 to 6 degrees Celsius. So this will have direct and indirect effects to the taxa that we're working with. So for example, there will be a reduced number of suitable habitats. It could be a loss of genetically unique populations under high warming conditions. It's been shown that in the Midwest, 70% of the 78 species will experience a range loss. And then warming waters can also accelerate development, creating this mismatch with food availability and altering life cycles. There can also be an increased variation in the timing of adult emergence, and this can create a greater risk from early to late storms and overall reduced population size. And there can also be reduced oxygen availability from higher temperatures, which create an oxygen debt. And so these are just some examples that will actually apply across taxa groups. And so as you might have noticed, mussels and crayfish are fully aquatic, and then also these 3 groups just have aquatic larval life, part of their life stage. However, across these groups, there's a lot of data deficient species, and that really limits our ability for conservation decision making, especially in relation to climate. So the Midwest Landscape Initiative evaluated 1,817 species of greatest conservation need, and these were species identified as needing the most conservation from state wildlife action plans. But the RSGCN process basically informs conservation planning across state boundaries. And so they found that there was a number of RSGN species for these different taxa groups, but aquatic species comprise nearly 2/3 of the RSGCN species. And so we're focusing on these 5 taxa groups, and ideally we wanted to include mayflies in this analysis too, but unfortunately, because of species level discrepancies and identifications, we had to omit them. So multi-species vulnerability assessments are a creative way to assess a large number of species that might be data limited. And there's a lot of different ones out there. And usually the output from them can also vary because it depends on the quality and quantity of data that goes into them and basically the overall objectives of the project. So we decided to use this Rarity and Climate Sensitivity Index that was created by Mims et al. In 2018. And since then, it's further been applied to fishes, amphibians, and reptiles in different regions. But what it does is it focuses on intrinsic sensitivity, which is basically a combination of species sensitivity determined using their area of occurrence, and then their adaptive capacity, which is looked at by adaptive traits. And then secondly, it incorporates exposure, which is the variability in climate across the species range relative to their spatial extent. So we had two main objectives. The first was to calculate relative indices of climate sensitivity for stream-dwelling invertebrates in the Midwest. And the second was to identify trait combinations that may aid in species adaptation to climate change. So we decided to focus on the Midwest Cask region. And this actually made sense as far as the data availability goes for a lot of these species. But after reading more documentation in the RSGCN and focusing on range-wide conservation for the mussels, we decided to expand the scope to include all of the RSGCN states and their extent. We used different methods of data acquisition for the different taxa, and this kind of depended on what experts were most comfortable in including as far as community science databases. We were fortunate enough to use two separate databases, one by David Houghton for caddisflies and Ed DeWalt for stoneflies. And then for crayfish, we used collections data and community science. Dragonflies, we used several different collections and community science databases. Mussels, we were fortunate to work with some state folks, so we were able to get their databases, and then we supplemented that with some collections and then iNaturalist data. So to calculate this index, we first needed to figure out what an occurrence point would represent. And so we decided that one occurrence might make the most sense if assigned to a small watershed. And that was a Huck 12 scale. So if one species occurred in 10 or more Huck 12s, then we decided to retain it in this analysis. And we only used data from 1970 to present, as this matched the climate data that we were using. We then combined all the Huck 12s for one species, and that basically was the species area of occurrence. And we overlaid these polygons onto climate rosters, which were selected by experts that they thought would basically determine species distributions. And then we would take the standard deviation of the climate throughout the species area of occurrence and standardize it along with the area of occurrence. And so by averaging all these climate rosters together, we obtained one value of climate sensitivity. And then by averaging the area of occurrence and the climate sensitivity, we obtained an index from 0 to 1, where 0 is least sensitive and 1 is most sensitive to climate. So we can look at these two measures that go into calculating the index. So here on the Y is climate sensitivity, which ranges from 0 to 1, where 1 is more sensitive, and the area of occurrence, where 1 is a larger area of occurrence, and the circles are standardized by the index value. And so here we're looking at this for dragonflies, and we can see that a lower area of occurrences, there's generally higher climate sensitivity. And if we look at this for crayfish, we can see that it's especially prevalent. So we have this really tight clustering. And if we look at the values, they're all between 0.75 and 1, or most between 0.75 and 1. So really high climate sensitivity and also very low area of occurrence. For mussels, there's this more variability in climate sensitivity, and then as you increase in area of occurrence, you kind of stays consistently at these lower values. And this relationship is also replicated for the caddisflies. The stoneflies are probably the most different in that even though they have this variability in low climate sensitivity, they actually still have higher climate sensitivity even as you increase the area area of occurrence. And so that might make sense because they are considered some of the most sensitive taxa groups and are often used for stream biomonitoring. So how can we apply this? We could basically assign these average index values back to the HUC-12 watershed scale. And in this way, we might be able to identify watersheds with a really high number of climate-sensitive species. So we can look at the richness that's found in these watersheds and then the number of climate-sensitive species within taxa groups. So this brings me to the second objective, which was to identify trait combinations that may aid species adaptation to climate change. And so while we did this for each of the groups, here's an example that we took for the mussels. So basically, we evaluated traits that may interact with climate to aid species adaptation to change. So the first step is to define a trait. So in this example, it would just be shell sculpture. The trait function would be that it might help the mussel anchor into the sediment better. And then the relationship to the environment is that maybe that will help the mussel withstand these higher flows. We then applied a functional niche concept where a species' ecological niche can be visualized as a multidimensional space. And then we can express these functional niches as probability density functions, which is basically the probability of observing a value for a given trait. And then we refer to these functions as trait probability densities, and these are calculated using kernel density estimators. And so, for example, if each of these probability density functions represents one trait, it's basically the probability of the number of species that have that value of the trait. And then instead of a histogram, we assign a kernel density so it looks smooth like this. And then what you can do is basically visualize this in a two-dimensional space and see where the trait combinations are most dense. So when we do this for mussels, we were able to use 60 of the 83 species in this trait analysis. We then used principal coordinate analysis to basically overlay on this trait density space so that we can see what traits are most used by different species. So here, if we look at the first two components of this principal coordinate analysis, and each of these eigenvectors are a trait, and we see that this heat map is showing the density of traits for different mussels. And we have this high density of traits here, which are relating to these two traits here. So a lot of mussel species use these traits. But what does this mean in terms of relating to climate? We could potentially relate these combinations of traits to our RCS index using a generalized additive model, because this allows for nonlinear relationships between traits and the index. So we can map this GAM over our trait space and see how it varies from lower index values in blue, which is less climate sensitive, to higher index values in red, which is more climate sensitive. And it's basically looking at the groupings of traits that might be related to these higher adaptive or lower index values, and then lower adaptive or higher or more climate sensitive. So then we have these trait combinations, and now we can assign them back to the mussels to see what mussels might have these traits. So here we're looking at an RCS index for mussels. So here are the species on the y-axis, and then the index from 0 to 1 going from least sensitive to more sensitive. And these dots here are color-coded by RSGC and status, and you might notice right away that there are a lot of RSGC and mussel species. So then these circles represent if a mussel has this combination of higher adaptive traits or lower adaptive traits. And so in general, this makes sense. So species that are more climate sensitive have these lower adaptive traits. But then also, some of these species at the top have these higher adaptive trait combinations. But then like species here, like Lampsilus cardium, is pretty widespread. It's considered tolerant, and it has these suite of traits, combination of traits, which are lower adapted. So what might be going on here is that even though this species has traits that make it susceptible to climate change, this species also has a really large area of occupancy. And so if we remember, intrinsic sensitivity is a combination of species sensitivity and adaptive capacity. And so we're really combining these two information processes because we're not using repeating the same information in each of these analyses. So what might this mean? We could identify potential adaptive and maladaptive capacity of species based on sensitivity trait relations. We might also be able to identify sensitivity hotspots or conservation priority watersheds around the region, and then perhaps identify species unlisted in the RSGCN process. So I'd like to think there's been a lot of people involved in this project who have contributed both data and ideas, specifically these folks in bold, and then also these people who have also contributed and just guided the project down here. So with that, yeah, thank you. Thanks for listening.
Great. Thanks so much, Isla. I'm gonna move the spotlight. So this